Now we will continue to our next speaker. It's my pleasure now to introduce to Sudip Kunwar, our Vice President. Sudip is a PhD student in the plant breeding graduate program at the University of Florida. He works with Dr. Ali Babar in the World Food Crop Breeding Lab. Originally, he's from Nepal, and he obtained his bachelor degree in the agricultural science from Agriculture and Forestry University in Nepal. Then he joined the Master of Science program at the Horticultural Science Department of the University of Florida in 2019. He worked with Dr. Jutta Albrecht, focusing on citrus physiology at the Southwest Florida Research and Education Center during his master's program and complete his degree in May of 2021. Then he received the Plant Breeding Graduate Initiative Award to pursue his PhD in plant breeding starting in the past fall of 2021. His PhD will focus on the improvement of wheat yield and physiological traits under heat stress conditions. With the use of UID-based hyperspectral imagery and genomics data, he will develop different prediction models for multiple traits using various regression and machine learning algorithms. Now he will share with us his presentation entitled Field Performance of Valencia Orange Trees on Diploid and Tetraploid Rootstocks in Different HLB Endemic Growing Environments. Welcome, Sudip.
Okay. Thank you very much, Jonathan, for the introduction. As you mentioned, my name is Sudip Kunwar, and today I'm going to present my master's research entitled Field Performance of Valencia Orange Trees on Diploid and Tetraploid Rootstocks in Different HLB Endemic Growing Environments. So here is the brief outlines of the presentation. I will talk about the introduction and objectives of the study, and then we'll move forward to the materials and methods. Then I'll be talking about the results and conclusion at the end. So citrus is one of the most popular fruits for flavor and nutrition, which belong to the Rutaceae family and known to be originated in Southeast Asia. It is cultivated in tropical and subtropical region, and US is globally top 5 citrus producing country. As per the 2020 data, Florida has citrus production of $729 million with economic impact of nearly $9 billion. So citrus has very high economic importance in Florida. However, a bacterial devastating disease named Wanglongbing, which is also known as HLB or citrus greening disease, has wreaked havoc the Florida citrus industry and reduced the production by almost 75% since its first appearance in 2005 from South Florida. So we can see some symptoms associated with HLB in the figure on the right. So most characteristic feature of this disease is blotchy mottle leaves, which is followed by effects on the fruit and flower size and tree decline, and at the end tree will die as the disease progresses. So as this study is about the rootstock, let's talk about a grafted citrus tree. A grafted citrus tree has two distinct components. The upper part is called scion, which is responsible for flowering and fruiting. And the lower part is called rootstock, which is responsible to attach plants to the ground and to absorb moisture and nutrients. These two components are joined at a point called graft union. Various studies have already shown different rootstock effect on the scion. Rootstock can impact on tree growth and production, fruit quality, and also it can help plant to develop resistance and tolerance against various biotic and abiotic stresses. So how rootstocks are useful on HLB mitigation? Well, most of the commercially important citrus scion cultivars, they are HLB susceptible, whereas some of the trifoliate orange hybrids mainly used as rootstocks have shown tolerance against the disease in greenhouse and in field condition, and also in graft combination with susceptible scion. Also, some studies have already proved that susceptible scion on the tolerant rootstocks were more productive, and they produced better quality fruits under disease conditions. On the other hand, rootstock can also be useful to reduce the tree size and to help growers to practice high density plantings, which has been demonstrated to improving yield efficiency and reducing cost of production for the growers, which can help citrus grower to compensate some of the losses caused by HLV with high efficient production. Therefore, demand for superior rootstocks has been grown tremendously. However, rootstock cultivar development needs long-term field study under multiple environments in combination with commercially important major scion cultivars. Also, the performance of rootstocks can be extremely different under HLB disease pressure than under normal conditions. So, extensive field study in combination with different scion under HLB disease condition was imperative. So the objective of this study was to investigate rootstock effects on horticultural performance of Valencia sweet orange trees grown in HLB endemic conditions. Materials and methods. So 2 trials were established in 2015 in the locations as provided in the map. Our first trial was located in Bessinger and second trial was located in Lake Wells. We studied 29 different rootstocks, which were both diploid and tetraploid rootstocks. And the design was completely randomized design with 6 replicated plots arranged linearly. There were 8 trees in each replications. So here we can see the differences between the 2 locations. The Bessinger trial was located in the flatwood type site with poorly drained sporosol soil, whereas our another trial, Lake Wells trial, was located on the ridge type site with well-drained antisol sandy soil. So rootstocks, here are the information about UF rootstocks, which were all tetraploid rootstocks developed either by somatic hybridization or by crossing at the tetraploid levels. And they were developed by UF rootstock breeding program at CREC Lake Alfred. Some of these rootstocks, which name starts with UFR, they are already available commercially. However, the one at the bottom here with asterisk symbols, they are not yet released commercially and they are still under evaluations. So here are other diploid hybrid rootstocks. They were mostly developed by the cross between mandarin and trifoliate orange hybrids, which is also called citrandarin hybrid rootstocks. And they were developed in California, Spain, or USDA, Florida. So the data we measured for this study were tree growth and production. We also evaluated fruit and juice quality. We measured leaf nutrient concentration, and we also monitored rootstock effect on tree health. We conducted one-way ANOVA analysis to test the significance of rootstock, and 2 kg SST was used to separate means and to test the significance among the difference in the means. And we also conducted a multivariate analysis. So results. Here we can see the 2 bar diagrams showing rootstock effect on tree canopy volume. The left diagram is for Bessinger location and right diagram is for Lake Wells location. So we have rootstock name in the y-axis here and canopy volume in the x-axis. Moreover, here 2 ploidy label rootstocks are distinguished by 2 different colors in the bar. That is red for diploid and green for tetraploid rootstocks. So rootstock effect for canopy volume was highly significant in both locations. And there was a trend that diploid rootstocks to produce a larger canopy volume than tetraploid rootstocks. The largest canopy, as we can see here, was induced by rootstock X639 in both locations, and the smallest canopy was induced by rootstock Changsha Benton in both locations. So here I have some field pictures. Here is the large sized Valencia trees. They were on diploid Spanish rootstock ES2. And here are some of the medium sized Valencia trees on Californian rootstock C22. And here are the small sized Valencia trees on tetraploid UFR6 rootstock. So they are all same as Valencia trees, but we can see they are like well different in their tree size. So orange were harvested in year 2019, 2020, and 2021, and the cumulative yield was calculated. In this bar diagram, rootstock effect on the cumulative yield is shown. Rootstocks are sorted on the basis of tree size induction. So here at the top, we have rootstocks that induce larger canopy volume, and here at the bottom, we have dwarfing rootstocks, just to see how tree size induction effect of rootstocks correlate with cumulative yield induction. So we found significant rootstock effect for the cumulative yield, and the trend shows here that the rootstocks that induce larger canopy volume also induced higher cumulative yield in both locations. Again, the rootstock X639, which induced largest canopy, also induced higher cumulative yield, and rootstock Changsha Benton induced smaller cumulative yield. But a higher yield per tree does not necessarily guarantee high revenue of a farm. And in that case, yield efficiency, which is yield per unit canopy volume, can provide more accurate information about the economic potential of a rootstock. So use of high yield efficient rootstock can increase the productivity of an orchard considerably if the planting density is optimized based on the size potential of the tree. So this is also important because recent studies have shown that HLB incidence was lower in orchards which practiced high density plantings. So we measured yield efficiency in our trials, and we found significant rootstock effect for yield efficiency at both locations. Here, yellow bars are for yield efficiency, and green bars are again rootstock effect on tree size, to see how tree size induction potential and yield efficiency of a rootstock associated. And we found that most of the rootstocks inducing smaller and medium sized canopy trees were more yield efficient. And as indicated by the arrows, these are top three larger canopy inducing rootstocks. And we can see that they were the rootstocks that induced below average yield efficiency in the trials. So total soluble solid is an important fruit quality parameter, especially for juice processing based citrus industries like that of Florida. And we found significant rootstock effect on total soluble solid content in the fruits in both locations. And the rootstock here on the x-axis, they are again sorted on the basis of their tree size induction from left to right. That means on the left we have rootstock inducing larger canopy, and on the right we have dwarfing rootstocks. And we can see that dwarfing and medium vigor inducing rootstocks induce higher TSS content in both locations. And the trend is more clear in Lake Wells location than Bessinger. So again, rootstock X639, which is larger canopy inducing rootstocks, induced lowest TSS content in both locations, and Changsha Benton induced highest TSS content. So interestingly, we were able to measure rootstock effect on trees' vulnerability to tropical wind. Hurricane Irma passed through both of our trials in 2017 and provided us opportunity to assess that effect. And interestingly, we found significant rootstock effect on trees' vulnerability to hurricane. So we can see that trees on here again, like we have sorted rootstocks on the basis of their tree size induction, and we found that dwarfing and medium vigor inducing rootstocks, trees on these rootstocks were more prone to leaning than trees on large canopy inducing rootstocks. We could not assess the actual reason behind that. However, it is more likely because of weaker root architecture of some of these rootstocks, and especially these two rootstocks, Changsha Benton C22 trees on these two rootstocks were more vulnerable to tropical wind-induced leaning, and might be a great trade to use them in a place like Florida, where tropical wind is an annual threat. So through the PCR, we found all trees were HLV infected. However, rootstock effect on CT value was not significant. Overall, we found only moderate expression of foliar HLV symptoms. It might be because rootstock were a little bit more tolerant to the disease and they induced partial tolerance, or it might be because we are still on the earlier early production years. So a rootstock effect for leaf nutrient was significant for some of the nutrients, but it was not consistent over production years and also over location. So we could not establish any significant relationship of any specific rootstocks to any specific nutrients. However, trees were overall were in optimum to good nutrient status, which is in contrast that HLV disease is known to affect nutrient condition of the trees. So here is the result from PC analysis for the selected variables. We used canopy volume, cumulative yield, yield efficiency, foliar disease symptoms, tree leaning, and fruit quality parameters. And we found that first two principal components explained 69%, almost 70% variations at Basinger and 64% at Lake Wells. In both locations, there was a separation of rootstocks based on the ploidy level along PC1. So diploidy was generally associated with healthier and larger size trees and higher cumulative yields on a per tree basis than tetraploidy. Whereas tetraploidy was associated with higher yield efficiency and better fruit quality. So this trend was more clearly defined at Lake Wells than at Basinger. So with these results, we found the diploid and tetraploid rootstocks evaluated in this study differed substantially in their influence on horticultural attributes of the grafted Valencia scion. Most of the tetraploid rootstocks induced a smaller tree size with high yield efficiency and better quality fruits than diploid rootstocks. Thus, utilizing small tree size inducing rootstocks in high density plantings may increase productivity and profitability in an HLV endemic environment, especially during the early production years before trees start to decline from the disease. So this might help the citrus growers to compensate some of the losses associated with HLV through early economic returns and also through cost minimization. With this, I'd like to acknowledge Citrus Research and Development Foundation for funding, Likes Bros for providing space for these orchards and taking care of them. I'd like to remember Dr. Bill Cashel and Dr. Fred Gimiter for their significant contribution in the establishment of these trials. And last but not the least, I would like to remember all my lab members of Citrus Physiology Lab, SOFREC, for their continuous help during this study. So with this, thank you everyone for your attention, and please let me know if you have any questions.
Thanks a lot, Sudip. Excellent presentation. So, and just a reminder to our audience, if you have any questions, please use the Q&A section. Sudip, one question I have for you is that if the differences you observe between the diploid and tetraploid rootstocks, do you think they are related mostly with the tolerance the differences to HLV, or more related to the nutrient uptake capacity and water uptake capacity of these rootstocks?
That's a good question. Thank you, Jonathan. So basically, the differences among diploid and tetraploid rootstocks in different horticultural traits induction, I don't think it's because of their tolerance against the disease. It might be partially, but it is more because of the differences in cell size and cell structure among the diploid and tetraploid cells, and the graft union response to the different differences in ploidy level among the scion and rootstock, because scion are all diploid rootstocks, cyan are all diploid cells, and rootstocks are either diploid or tetraploid. So maybe discrepancies in ploidy level between the cyan and rootstocks has major role in limiting the growth and inducing the effect differently than their effect on the HLB.
Thank you, Sudip. Also, when you are doing the grafting between using a diploid or tetraploid, which one is more susceptible to fail and become a successful tree at the end?
Thank you. So basically, this trial were established in 2015, some years before I joined for my master's, and I didn't do any grafting with these rootstocks and cyan. But of course, I think it is more difficult to graft tetraploid and diploid cyan and rootstocks than both diploid components. So I think I don't have any exact figures right now, but I'm sure that it will be a little bit less successful, and the success rate is a little bit high when diploid cyan is joined with diploid rootstock.
Thank you so much, Sudip. Excellent presentation. Thank you everyone for being with us one more time again with us. We will have our lunch break until 1:50 Eastern time. After that, my colleague Swati will be introducing our third and fourth student speaker after lunchtime. So I'll see you all at 1:50 p.m. Thank you.